Okay, so hello everyone. I think we are now ready to start. Once again, we are sorry about the time zones issue. So thank you for still being connected to the meeting. And today we will talk about single cell analysis, and we will go a little bit into the detail of the analysis, and we will also take a look at the programming behind the analysis. So I will now share the slides, and you can tell me if you can see them. So, first of all, I have to present, because today I will be the only one boring you, but other people contributed to this tutorial. So I am Laura Veschetti, and I am a postdoctoral researcher. Just now I got to be a young PI on a project, and I am working in San Raffaele Hospital and University in Milan. Then we have Mirko Treccani, who is also connected today and will be my friend, who is also a postdoc at the University of Parma. And then we have Professor Giovanni Malerba, who is a full professor in medical genetics. Just a brief disclaimer, it could be that you hear some alarms or motorbikes because I live near a very busy street, so I'm sorry in advance for any disturbance. In case you cannot understand me, just write in the chat and I will repeat. So, as I said, we will go through some analysis regarding single cell. First of all, we will go and have an overview on the single cell RNA sequencing in general, and we will see an overview of the workflow. We will go then seeing a little bit of the programming, so in R and RStudio, and then we will go and have a theoretical part again, let's say, and that part will focus more on the clustering workflow. And that is what we will be doing again in code in the hands-on session 2. And then at the end, we will try to conclude very briefly with a recap of the concepts, and I will give you just some hints about downstream analysis. So where do we go from what we obtained today? Just for your information, the code I will show you today and we will go through and understand will be loaded in the shared folder right after the tutorial. So you will have all the code with the comments so that you can go through it also alone. So we can start by delving into single cell RNA sequencing, but first of all, I want to propose somehow a brainstorming, about what do we mean when we say cell type. What are cell types? So if you are a pathologist, for example, you would, for example, show me a slide. This is a hematoxylin and eosin stained colon slide, and you can just show me that the tissue has a different architecture, and then we can see different cells that at the eye have different characteristics, let's say. Whereas, for example, if you are a bioinformatician, you could propose me something like this, no? So we have, for example, many expressed genes, and some of these genes are shared among cell types and characterize a group of cells when compared to all others. But in this case, we do not have, let's say, maybe a physical representation, no? Of what does it mean. And I think that single cell is a very nice way of combining these things. So when, for example, we will see how to generate these graphs. These are UMAPs, and each dot on this graph represents a cell. So a cell with all the transcripts that are contained in it. And for example, in this case, in this nice paper, they somehow highlighted some cell types by some markers, and they performed also the immunohistochemistry. So you can have somehow the, I don't know, virtual, let's say, representation of cell types, and you can also see actually to what it translates. And why is this important? Because sometimes, for example, in this slide or in this slide, you actually cannot see any phenotypical difference between cell types, but they are actually expressing something different than when compared to all other cells, no? So I think this is a very interesting point of view. And one could ask, yeah, but why do I have to study each cell type in a specific way, no? Each single cell. Here I reported just a couple of slides reporting tissues and organs that can be found on the Cell X-Gene database. This is a database for single cell data sets. Okay, so we have here the tissues and the organs, and here the cells that have been identified as belonging to that tissue or organ. And if you can see, there are many different tissues and organs, but I took as an example the blood. If you open that tissue, you get all these different cell types. Okay, and these are not just different cell types, but sometimes they are different cell states in that cell type. So, for example, we can see, for example, here, activated CD4 positive T cell. So we have different T cells, and in this case, these gives us also an information about the functional state of the cells. So, as you can see, and this is just the blood, no? As you can see, we are actually very, very heterogeneous in terms of cell types and states in just our organism. And spoiler, we are not the only one, of course. And so it becomes very important to get a message that is specific to each cell type. So to somehow get insights about the functionality and the state of each different cell types. So here I reported some reasons, because when we set out to perform a research or a project, a study, there is a point in which we have to ask ourselves which is the best method to see what I want to see, no? And of course, a single cell is a very advanced method that gives us much resolution. But as we will see in a few minutes, sometimes there is also a balance between the resolution and the message that I want to see. Okay, so why choose a single cell RNA-seq? We choose this method, this technique, when we want to explore, for example, which cell types are present in a tissue, we want to identify some unknown or some rare cell types or states, so a state of activation or not. We want to maybe elucidate the changes in gene expression, and this can be during, for example, the differentiation process, or, for example, it can be when we have a control group and somehow a treatment or an exposure to some kinds of stimulus, okay? And it is very powerful because we can see the effect, for example, of the treatment in each cell types, okay? So here, just to balance all the methods out, I reported some comparison that we can draw between the bulk RNA-seq and the single-cell RNA-seq. And then we will see also a comparison between the single cell and the single nuclei RNA-seq. So bulk RNA-seq, let's say, is maybe the most known, the classical way, let's say, of studying RNA and expression. And here, when I thought about it, I found 5 main key points that one has to somehow evaluate when choosing which technique is the best for the project, of course, and for the study and what we want to see. So, for example, what in terms of resolution? Bulk RNA-seq gives us information regarding the average expression of transcript in all cell types that we are studying, whereas the single cell captures the expression at individual cell level, and so we can have information about cell types. Cellular heterogeneity, bulk RNA-seq cannot detect cellular differences because we have a mixed signal somehow, whereas single cell resolves heterogeneity between cells, and we will see later how to do that. Regarding the application, for the bulk, we can have a comparative transcriptomics or some disease biomarkers discovery. And usually, what do? We perform a differential expression analysis between one condition or another. Okay, whereas single cell can have more variety, let's say, in the applications, because, as I said before, we can do the comparative transcriptomics, the differential expression analysis for each cell type that we are studying, but we can also use this data to find new or rare cell types. So, cell types identification. Then, what about the complexity? The bulk RNA sequencing is, I say, straightforward just because it is maybe more known, and it is many years that we have a standardized analysis pipeline for analyzing this data, and it is cost effective, so it does not cost that much. Whereas a single cell, we have more information, more data. So we also have somehow computationally demanding analysis, and we have a higher cost. So we have, let's say, more information and more resolution, but it comes at a cost, which is computation and money. Then what about the output? The bulk RNA-seq, we get global gene expression profile. We can do, for example, pathway analysis and stuff like this. Whereas for the single cell, we have this information for each cell and for each cell type. Okay. So here I reported a graph just to explain better what does it mean that we have average expression and when we have expression at individual cell types, no? Because think about the graph on the left. Here we are, for example, in the setting of a bulk RNA-seq. No, we have the expression of the two genes, okay, gene A and gene B. And if I analyze a heterogeneous group of cells, okay, and I want to see the correlation between these two genes, the calculation would return something like this for the correlation score, no? Whereas if I am analyzing the single cell, so the graph on the right, and I actually get that this group is heterogeneous, so it is actually made up of a group of cells. There are the green cells and the blue cell types. I can actually unveil the true correlation between these genes, so the gene A and gene B, which is actually a positive correlation in both cell types. Okay, so in this case, we have the data, information, and resolution so that we can get true signals that are actually specific for each cell type. Okay. Yes, what I just told you, of course, does not mean that one method is strictly superior to the other. In fact, very recently we published a work exploring the exercise-induced transcriptional dynamics in runners. Okay, they run a half marathon. And we perform single cell analysis. But in our work, actually, we also we performed the classical single cell analysis. So we went and saw each different cell type, what it happened in them. But we also did some kind of methodological aspect, let's say, that actually has a biological relevance, in which we treated our single cell data and we put it all together as if it was a bulk RNA-seq, okay? And we perform differential expression analysis also in this way. And actually what we found was that analyzing this data also as if it was a bulk RNA-seq experiment gave us complementary findings. And this is especially true when you are working with few individuals, okay? Of course, as regards for example, the pathway analysis part, we have, as you might expect, more noise when we analyze it all in bulk, whereas when we analyze it by cell type, so using the single cells, we have a more clear picture. But what about the single genes? Somehow, and sometimes when you put all cell types together, you can see trends emerging. So maybe in a cell type, there is a small variation in gene A, which is overexpressed post-running. Also, in cell type B, it happens the same, but this is actually so small that we do not see it as significant, for example. Or maybe also the cell type is very rare. So we have very few numbers of cells belonging to this cluster, no? Somehow, when analyzing all of them together, we can boost the signal of these genes, and maybe some of them become significant. So it could lead, let's say it is an exploratory way to find some markers, for example, no? And then you, of course, go on and validate it. Then I told you before about the single nuclei. So what is the main difference we are talking about? In both of them, we have a single cell resolution, okay? But in one case, we analyze the whole cell. So we have the cytoplasm and the nucleus. Whereas when we talk about single nuclei, we consider only the transcripts that are present in the nucleus. And this is done mainly when there are tissues that are difficult to disgregate, or maybe we have frozen tissues or FFP tissues or slides. And the main difference is that when we analyze single nuclei, we can have transcripts that are actually just in the nucleus. So we also expect to have maybe introns, okay? They have not been spliced yet, so they are pre-mRNA. Whereas when we analyze the whole cell, we expect to have more mRNA in respect to pre-mRNA. And this is important, and we will see it later during the workflow, because you have to decide whether to keep or discard the intronic reads that we see, okay? And also one limitation of both methods, when we are working with single cell, usually we can also see that there are some procedures to extract particular cell types. So for example, I am analyzing the blood, but my study focuses on some rare populations on the T cells. I might want to just analyze the T cells, so somehow isolate them. But this is usually done with the FACS, so the cell sorting, no? And these techniques have been demonstrated that can somehow stress the cells, so it can be that their transcriptional profile get altered, okay? Whereas the single nuclei, this does not happen, because you make literally the cell explode and you get just the nucleus because it has a stronger membrane, okay? And on the other part, we have fewer transcripts, because once DNA gets transcribed into our RNA, it is usually put into the cytoplasm, okay? So, as I said, very brilliant method, super technological, but there are some challenges. Otherwise, everyone would be doing it with closed eyes and not ever thinking about it. Here I reported 4 main challenges, which is the large volume of data, the low depth of sequencing per cell, biological variability, and technical variability. So, what about the large volume of data? Of course, when you are performing an RNA-seq experiment, single-cell, RNA-seq experiment, in addition to a lot of money, you have to keep into account that you will require high memory to process these very large data sets, storage, in order to keep all the raw data, but also maybe some steps of the analysis, because as we will see later, some steps of the analysis, if you have many cells, require a lot of time. So you would run them one time and save the results. Otherwise, you have to run them all the time. It will take too much time, okay? And of course, longer analysis time, because the more information I have to analyze, the more time it takes, okay? And another point, the computational resources are very critical. So just you have to put it in your bag, let's say, that for processing the raw data and obtain, as we will see in a few slides, the count matrix, you will most certainly have to work on a server, okay? Or you have a very good computer with a lot of RAM, otherwise you will need some form of high computing service. The other very important point is the low depth of the sequencing per cell. So it is true that we generate a lot of data, but for each cell, we have actually a shallow sequencing. So we actually managed to capture and this also varies according to the cell type, how much variety of transcripts are they transcribing, okay? We only detect up to 10-50% of the transcript per cell, okay? So as you can imagine, if one gene is lowly expressed, I have very few chances of sequencing it. So we end up with these many huge counter matrices which are full of zeros, and this is for this reason. So we have, first of all, not all the genes are expressed in a given moment. So we can add zeros for all the genes that are not expressed at the moment. And then we also have to add zeros. Why? Because we lose some genes. So we have a very this varies according to the type of cell, but we have a chunk of genes that are lowly transcribed, so lowly expressed. These probably we will not get with the single cell, so these also are zeros, okay? So we end up with these huge matrices full of zeros, and then we will see later how to manage it. Then, of course, we have different expression patterns. So genes with higher expression have fewer zeros just because of this, and some genes are not detected in any cells just because they are not expressed or they have a low expression level. We have then another big problem, which is variability. On one side, we have a biological variability. So we have somehow sources of unwanted variation that could lead us to some messages that are not actually correct. Okay, or they can mix things up and make some noise, let's say, that will not allow us to see a true biological signal, no? And some examples of these are, for example, transcriptional bursting, which means that genes are not always actually transcribed. So the time at which we collect a cell also determines whether a gene is expressed or not. And then there is also, for example, you can think about cell cycles genes, no? So, these cyclins are expressed at different time points during the cell cycle. So just by getting a bunch of cells, and if they are in phase S, M, they have different genes expressed, but that would not be a biologically significant thing for us. Of course, it depends on the type of study, okay, the aim of the study. But for example, if we are comparing treatment versus control, and just by chance, in the treatment, we collect more cells that are in the S phase, in the controls in the M phase, we would see that some genes are differentially expressed in a significant manner, but actually it is just the cell cycle, okay? And another example is given also by the RNA processing rates, in which different RNAs are processed at different rates, just because of the length of the transcript, but also on the turnover, for example, of the protein and stuff like this. Then we also have the problem of the continuous or discrete cell identities. So, for example, we have for the T cells a different degree of pro-inflammatory potential, and this is on a continuum. And the fact that it is a continuous character makes it difficult to divide them somehow and binarize the phenotype. So it is 0 or 1, pro-inflammatory or not. It is very difficult to distinguish these phenotypes because there is a gradient actually, no? Then we have the environmental stimuli. So we have the presence of hormone and signaling molecules and stuff like this that can change the transcription. Then we have also temporal changes, and it was what I was saying before. So we can have altered gene expression just because we take the cell in a different phase of the cycle. Then we have technical variability, which is mainly linked, as it says, on the methods and on the technique. So we can have different cell type specific capture efficiency. So just by chance, some cell types can be more analyzed than others. The library quality, so we can have degraded RNAs, dying cells. For example, neutrophils are known for dying very quickly. So if we are analyzing PBMC from blood, neutrophils are the first one that die, okay? So if we are not very fast in getting everything, we will not have any signal regarding the neutrophils. Then we can also have amplification bias or the batch effect. The batch effect, I reported here a very nice representation because I think it is a very important thing that we have to remember. We do not have, when we are setting the design, we cannot confuse the batch, the processing batch with the biological group. Because in this way, which is the upper part of this graph, we end up with differences which we cannot say whether they are true biological differences or it is due to the batch effect, no? Whereas if we always split up the biological groups and process them in mixed batches, we can actually say whether it is a true biological difference, so the triangles are different from the square, or actually if what we are seeing is a batch effect. So as you can see, all the colors represent the batches. So all the orange samples cluster together, but it is not due to the biological fact, but due to how I process them, okay? So I think this is very important. Then I will give you a little bit an overview of the workflow, just so we know what we are focusing on. And I will start briefly to explain to you this initial part that we will not see computationally, but because it would take too much time, but is always important to know which are the logical steps in the workflow. So first of all, we have the sequencing and alignment. We have the gene quantification, and we will see just now what this is. Then there is this big part, which is what we are actually doing today, also computationally. And we have a fourth step of quality controls and cell filtering. We have count normalization, feature selection, dimensionality reduction, visualization, and clustering. And this is all that we will go through today. So you will end up knowing what every terms in this part of the workflow means. And then we will just give hints about the downstream analysis, okay? One important thing that I want to tell you is that it does not matter whether you sequence millions of cells from one sample. If you want to make conclusions about your study that corresponds to the population, so treatment versus control, for example, you need biological replicates. Otherwise, you are just saying something relating to one individual. It doesn't matter if you are analyzing one cell or 1 million, okay? So just for your knowledge, today we are focusing on this part. So this is dependent on the library part. methods that sequence the 3' end of the reads, which comprises all the droplet-based method. And why this? Because they are the most popular methods. For example, the 10x genomics, machine and library preparation, they are all 3' end reads, okay? So be careful when you are actually doing this part about the library preparation. So you can expect whether it is the 3', 5', and stuff like this. So I just want you to show the library construction, because in this case we are sequencing in per end. So we have a read 1 and read 2. We have the P5 and P7 adapter for the Illumina sequencing, okay? Let's focus on read 1. We have the read 1 primer, which is where the sequencing primer will attach. And then the first read actually is just a read that tells us information regarding the belongingness, now I will explain, of the transcripts. So we have a sequence that is the 10x barcode that gives us information regarding to which cell the transcript belongs. So we have one barcode for each cell. And then we have the UMI, which is the unique molecular identifier, which is a sequence that is different for each molecule. So in this case, this gives us information whether this is a single molecule or a different molecule, okay? And then here, if it goes through, it should not, but if it goes through, we will find the poly-DT. The read 2, instead, will give us information on the insert, so on the 3' end of the transcript. So the read 1 actually is just information to see this transcript comes from this molecule coming from this cell. And the second read actually contains the transcript, so we can see which is the gene that is expressed. Then here we just have the index in case we are multiplexing, so analyzing multiple samples in just one flow cell, okay? So this is an example for the UMI. So for example, these two reads, okay, first of all, we have different colors, as you see. These are the barcodes. So these red barcodes means that it comes from cell 1, blue cell 2, and so on. In this case, the UMI is important because we have two reads that come from the same molecule, okay? So if they have the same UMI, they actually should be collapsed. Otherwise, we are counting 2 times the same thing because it is the same molecule. Instead, if we have different UMIs, they are 2 transcripts, both mapping on the same gene, okay, on the same transcript, but they come from 2 different molecules. So we should keep the count at 2, okay? This is the importance of the UMI. Then here it is a picture coming from the 10x genomics documentation. They have very long and detailed manuals and protocols, and these just to tell you how the first part works. So usually you have the cell samples, you use the Chromium chip, which has many wells to do the library preparation. So in each droplet, we have one single cell, and for each cell, we have a library, basically. So we attach the barcode and stuff like this. Then we can take multiple samples and put them together. So we prepare the library individually, and then we put them together on the sequencer. We sequence everything on a single flow cell, for example, in Illumina. And then we have this software, which is Cell Ranger, which is particularly fitted for the analysis of 10x data. And this tool allows us to make, for example, the conversion between the BCL to FASTQ. So we obtain actually the FASTQs Then we map everything and we obtain the counts, and then we can aggregate the counts coming from each experiment, okay? So here, just for your knowledge, I reported here that there can be different kinds of reads, so they can map on different parts. They can be exonic reads, for example, that map only on the exons, intronic, which map only on the introns, transcriptomic, that both map on the exon and on the intron. And this is the case, for example, of the single nucleino. We are analyzing pre-mRNA. And then we have some antisense read that are actually oriented in a different way, which is actually wrong based on our library construction. So here I will not go into the detail of this, but just for your knowledge, we map all these reads on the referent genome transcriptome of the organism. And then we apply some filter. They are quality filter, for example, for the mapping quality, but also we can choose to discard, for example, the intergenic or intronic reads based on what we are analyzing. We then keep the good quality reads, and we can then perform the UMI correction, because sometimes the UMI gets read wrong, but maybe only just one nucleotide. So there are some statistics software to correct these UMIs. And we have then cell calling, in which a tag is added to tell us whether a transcript comes from a cell or another, okay? And the result of all of this is the count matrix, which is the input for our analysis, okay? Which has as row the genes, and as columns we have the cells, and we have the expression level of that gene in each cell, okay? So we have these very huge matrices. And up to here, I will stop now and give you like 5 minutes break, just because I gave you a lot of information and I want you to digest everything. Then we will go into the hands-on part, so we will see a little bit of the analysis. And of course, if you have any question, you can write them in the Q&A box, and we will answer them after the break. So we will resume in 5 minutes. Okay, so I think we can resume now with the hands-on part 1. So I will now share the screen again. Okay. Okay. So just before going to the coding part, let's say, I want to give you a very brief overview on what we are going to do. So first of all, you should have been able to download everything from the shared folder on the drive. In there, you have all the data to perform this analysis. And as I said before, after we finish the tutorial, I will also load the code that we will see. Then, now we are going to see an overview of the data set. So with the experimental setup, where these data come from. Then we will perform some quality controls and we will filter our data set. Okay, so this is just a different way of showing you the workflow from before. So this one is the one regarding the sequencing up to the expression matrix. And then everything we are doing today is from the expression matrix onward. So right now we will see and we will prepare the data set. We will choose the cutoff for the quality of our data set, and we will filter our cells and transcripts according to some thresholds. But first of all, I just want to give you a little background on the data we will be working on today. Here I reported the reference to the work that published it. And in particular, the work actually was aimed at distinguishing single cell from doublets, which is what? If you might remember from before, when I said that the libraries are formed in a droplet containing a single cell, it could happen that in this droplet, two cells end up, or other droplets might be empty, no? If in a droplet two cells are embedded, the library will call all transcripts coming from these cells as one single cell, coming from one single cell. And this is the doublet concept, okay? But we will not focus on that today. We will actually go and look at some biological understanding of this data, because these two data set, they come from PBMC, so peripheral blood mononuclear cells, and they come from 8 lupus patients. We have a data set for the stimulated, which was treated for 6 hours with interferon beta, sorry, and the control data set in which these cells were left untreated. The library preparation is the 10x genomics, so the one we saw, and one of the most popular ones. And these libraries were sequenced on the Illumina NextSeq 500. Here I left you some references for the GEO and SRA databases. So if you want to download the raw data from this publication, you can. And some metadata regarding this. We have two pooled groups, the controls and the stimulated. They both have around 13,000 cells. And since we are working with PBMC, we will expect to have some immune cells in our sample. And we can expect to have B cells, T cells, NK cells, monocytes, and so on. And in this case, it is highly important to know which kind of cells do you expect to be in your data set, and also the somehow numerosity of your cells. So the how much are these cell types different? Do you expect a great variability or not? Do you expect a low complexity or high complexity transcriptome inside of your cell? Again, do you expect to have high mitochondrial transcripts or not? And this is very cell type dependent. And based on the cell types you expect to analyze, you set different thresholds for your quality control filters. So, as I said, set expectation. Here we do not expect to have very high complexity cells, nor very high mitochondrial content. So we will somehow exclude cells which do not fit our expectation. Now we will actually switch to the code. So I will change the screen. Okay, I don't know if you can see RStudio. Okay. Okay, so first of all, you need to be into the folder of the data that you downloaded, or you have to set the working directory in R. Otherwise, we will not see the data. So in the folder that I put on the drive, you will find a Seurat integrated object that we will see later, and we will use it. And you will see two different, sorry about the ambulance. You will see two different folders, the control and the stimulated folder. Each of them has three files inside. We have the barcodes file, which is a text file which contains all cellular barcodes present in that sample. And these are ordered so that when we use the three files, we actually link this information to the count metrics. And so they are in a particular order. Once again, okay. Then we have the feature file, which is a text file which contains the identifier of the genes. So the cells and the identifiers of the genes. And in particular, the identifier, of course, can be different based on the database on which you annotate. So we can have the ensemble ID, the CBI ID, USCSC, and so on. So you need to be very careful about which identifier you are using, because when you then link the annotations, it can be quite a mess if you are trying to get annotation from a wrong key. Okay, you will not get anything at all. Once again, the feature, in this case, they will become the rows of our matrix. And then we actually have the matrix that contains the count values. So just as the slide I showed you before. Of course, as I said, full of zero values. So first of all, we have to load the libraries. And if you had any problems installing these libraries, I actually put a README with all the references to go in order to troubleshoot the installation. So you should be able to install it. And then we actually go on and load the data. So I will start it so that it can load in the meantime. So in this case, we will use the read tenex function because we are dealing with the tenex data, okay? And in this case, we do not to specify each single file, so the barcode, the matrix, and so on. But we will just specify the directory name, because in automatic, it will take these three files and combine them together. So, So as you can see, we have generated some Seurat dataset. The output of this function, so the object that we obtain, as you can see over here, they are the Seurat objects which have a very specific architecture, okay? And for example, if I just open, you can see many different voices, and the more you perform analysis on this, the more these objects will get layers and information packed inside, okay? Okay, so, okay, also here we can select the mean feature arguments, which specify the minimum number of genes that have to be detected per cell. So if we have cells which have less than 100 genes, we will discard them from the import start. And that's it. This is just a sign what we are getting to this to cycle, okay? Okay, so as I said, Seurat object. The Seurat object stores all the information associated with the dataset. And as I showed you before, there are different slots which contain different kinds of information. And usually they can be accessed through a @ symbol. So you can use the name of your dataset, @ and the slot name, and we will see some of them during the analysis. So for example, we have a slot for the metadata, we have a slot for the RNA row counts, we will have a slot for the normalized counts, and so on. And for example, I can show you here the metadata layer of the Seurat object for the controls. So you can see here is the cellular barcodes, which is encoded in the orig ident. Then we have the N-count RNA, which is the number of UMIs per cell. And then we have the number of feature, which is the number of genes detected in that cell. Okay, and the same we have for the stimulated, so the treated with the interferon beta dataset. Okay, so of course now we have two different objects, but we want to work on them in a unified manner, so we can merge our dataset. And as you can see, we use the function merge the dataset, the Seurat object X, which is the control, Y, which is the stimulated, and then we also add the cell ID as control or stimulated. Why this? Because it could be that some barcodes are used in the are both present in the two experiments, okay, in the two condition datasets. So we would not want to somehow confound these, so we differentiate, okay? Then we concatenate the count matrices of both samples. So we join the layer, okay? And these can be seen, so we are now working with the merge Seurat, okay? If I put head, I can see all the controls. You can see that now we have control and the barcode, and the tail of the same layer, so the metadata, is everything is stimulated. So I attached at the bottom the other dataset. Then what about quality? We need to start to explore our dataset, no? First of all, we can somehow compute a score, which is the novelty score, which is the number of genes detected per UMI. And this gives you an information about the complexity of the dataset. So, for example, if we have many UMIs that all call a single transcript, we have no diversity, we have only the information that one gene was present. So maybe this gene that was highly expressed somehow pushed down the signal for all other genes, no? And we will only see that. And in this case, it means what? That we should have sequenced more, okay? So we can add the number of genes per UMI on each cell, okay? For each cell, we calculate this and we put it in the metadata. And as you can see, we just divide the number of features, so the genes, divided by the number of UMIs. And this is done just in log10, just to not have a huge variety of values, so a huge range of values, okay? Then we have the mitochondrial ratio, which is another very important parameter, because in this case, it gives you the content of mitochondrial transcript in the cell. And why is this important? Because if we do not expect to have a high mitochondrial content, and we see it might mean that the cell is in stress, Okay, so it upregulates all the mitochondrial transcripts, and it is somehow in stress and maybe going to die. Okay, and we will not get meaningful results from this. So again, we calculate the mitochondrial ratio. So we have here a function which is a percentage feature set, which is in the Seurat package, which automatically gets the pattern that you specify. So here we are looking at transcripts that start with MT, which stands for mitochondrial in human. And here, just a note, be very careful about the uppercase or lowercase, because, for example, I worked with mice data, okay? And in this case, MT is lowercase. So you need to be sure in your organism how the mitochondrial transcripts are somehow identified. Okay, so here we will look for the pattern MT in the transcripts, in the object merge Seurat, and it automatically calculates the percentage of the feature with MT, so the content, and we will store it in a column which is called the mito ratio. Okay. Then, since we are actually more, let's say, at ease, not with percentage values, but without the 100%, we just divide by 100, okay? Okay. Then, we now have to work with the metadata, okay? So instead of working with the entire object, we can extract the metadata and work on it, make our reasoning, and then merge back the metadata on the object. And this is just to make sure that by mistake you modify something else on the object. This is just a tip. Of course, when you do the analysis, you can do whatever you want. I find this to be very useful to avoid the mistakes. Okay, so in this case, I take the metadata layer from the merge Seurat and I put it just in a metadata dataset, okay, data frame. Then we add a new column for cell identifiers, okay, and we do this. Why? Because currently the cell identifiers are in the name, okay, in the row names of our data frame. And we can do this by getting the row names of the metadata, and we put it in a column which I call the cells, just to remind that this is the cell identifier. Then, just to give you an overview of what we have right now. So we have row name, count, so the UMI, and features, which are the genes. This is what we calculated, so the novelty ratio, so log 10 genes per UMI, okay, this is the score that we calculated above. This is the mitochondrial ratio content that we calculated. And then now we have a column with the cells, okay, which are the identifiers, okay? Okay, so here we can also use the fact that we attached the control or the stimulated cells to the name of the cell barcode to generate another column which contains the group of our sample. So I generate an empty column in the metadata, which is called sample, okay? Then I will take the ones that start with control, so these ones. I will get their position, and in their position in the column sample, I will add the control. Whereas when there is the pattern stem, in the sample column, I will add stem. Okay, and just to show you here, as you can see, we have a column sample, and if we go to the end, we should have stem. Okay, so we have information about the groups. Then we can rename some columns, for example, the n count and the n feature, so that we don't have to remind every time that these are the UMI and these are the genes. So I can use the dplyr. So the column, which is the n count RNA, now will become the number of UMI, and the n feature will become the number of genes. Okay. Then we merge back the metadata to the Seurat, and then always very, very, very important, we can save the Seurat object, so that the next time, if I have to interrupt here the analysis, tomorrow I open the PC, I just load the object, and it is all fixed with every calculation, okay? So you can save, and it will save the object wherever you put it, okay? Then we can start assessing the quality metrics. Yes, metrics. So we start analyzing the cell counts, okay? So how can we estimate the cell counts? We can estimate the cell counts by counting the unique cellular identifiers, which is given by the barcode, okay? In this case, as I told you before, we expected around 13,000 cells. And we can see whether we analyzed less cells, for example, or there could be a case in which we have more cells than expected, and in this case it might mean that some of them are actually not cells. Okay, we sequence something else. So here, what did I do? I took the metadata data frame from before, and I plotted by sample. So here, tell me if this is too small, but I can see it for now, and then maybe I will make it bigger. We have the control group and the stimulated group. And here I just plotted the count of the barcodes. So we can see that we have more than 15,000 cells. So probably some of these barcodes do not actually indicate some cells, but maybe they were just free RNA that somehow got into a droplet, okay? And we are seeing them as cells, but they are not cells. There are now some methods to estimate whether it is truly a cell or not, okay? And to do this, we can plot, for example, the UMI counts. Generally, we should expect to have more than 500 UMIs per cell. Otherwise, we have really too few information. And this could mean also that these are not cells, but they are just RNA floating around, okay? So here I have the cell density, and here we have plotted the number of UMIs. Here there is a line, which is this one, which I set at 500, because 500 is the minimum threshold that I want. So in this case, we are pretty happy, because both in the controls and the stimulated sample, we have the great majority of our cells that have a good number of UMIs. So we would end up only discarding these fractions of cells, okay? Then we can also visualize the genes detected per cell. So I will plot this. Okay, here, same idea behind the plot, so the density, and here we have the number of genes. Usually we consider a threshold of at least 250 genes per cell. Same reason, if I see less, it means it is not truly a cell, okay? Once again, we are happy because the majority of the cells end up here. And also the shape of this graph could somehow give you a little hint about cell population present in your sample. Because if we have, like here in the control, we have almost 2 peaks, no? It seems almost a bimodal distribution. And this will mean that there is a subpopulation which is more rich in transcription. If, for example, we would see maybe a shoulder here, it would mean that maybe some cells are quiescent. I don't know how to pronounce it correctly, but they are not very active, let's say, okay? So just by looking at this graph, we can get some information about the heterogeneity in our subpopulation. Then we can also plot the novelty score, which is the one that we computed before, okay? Same thing, so density, these are the thresholds. Here, be careful, we are talking about a logarithmic, okay, measure, because we perform the log 10 genes per UMI. And here we have somehow a measure of the complexity of our cell transcriptome, as I said before. And also here we are very happy because the great, almost all cells in our samples have a good novelty score, okay? For example, in our data set, we could expect that maybe here we could see maybe some red blood cells, some reticulocytes, no, that have some transcripts left in them, but they are just hemoglobin transcripts, okay? So they are very poor in the novelty score, okay? Then we can also plot the mitochondrial ratio. So same thing, density, mitochondrial ratio, but here, of course, we do not want that our data is above the threshold, but that is below, okay? So also in our case, we are very happy. In this case, we take, but again, this is very linked to the kind of cells you are analyzing, okay? Here I put a threshold of 0.2 for the mitochondrial ratio. But for example, if you are studying, I don't know, muscle cells, you expect that there are many mitochondrial content. So maybe this threshold is a bit too restrictive. So you have to find a balance according to the cell types we are studying, okay? Then, up to now, we only saw plots representing one measure, no? But we do not know if, for example, the cells having a low count of genes are the same having low genes per UMI or, for example, low mitochondrial content. So it is always better to join these data and look at the distribution of different values. And here we have a visualization, okay, of these, maybe a little bit, okay. of these. So on the x-axis, we have the number of UMIs, okay, here and here. They are divided by controls and stimulated. Whereas here we have the number of genes on the y-axis, and we have a color gradient that indicates the mitochondrial ratio of the cells. So the more it is black, leaning toward black, the more mitochondrial transcripts we have in our cells. So you can see that we have these cells which have big number of UMI and a great number of genes. Low mitochondrial content because it is light gray, okay, so it is close to 0. So these are good cells. They are cells from which we can take all our information, whereas these cells, they have a low number of UMI, they have a low number of genes. Some of them, also here maybe it is more visible, have a high mitochondrial content. So this could mean that either these are not cells or they are cells which are going to die. They are too stressed and they are going to die. So the only thing that they are transcribing is mitochondrial contents, or also that they could go toward necrosis. So mitochondrial pop and release all the transcripts in the cytoplasm, okay? So we took a look at this. Of course, these are the same thresholds as before, okay? And based on this and on the population that we are analyzing, we can choose some thresholds. In this case, since we are working with PBMC, I chose a threshold of number of UMI of 500, number of genes greater than 250. The novelty score must be greater than 0.8, and the mitochondrial ratio must be less than 0.2. These, of course, are somehow, let's say, subjective thresholds, okay? But the numbers I give to you, these ones, are actually the starting point, okay? Then, based on the type of cells that you are analyzing, you can adjust the thresholds. And these are very important because based on this threshold, you decide whether to keep or discard a cell for the following analysis. So if you are too relaxed in these thresholds, you end up with many junk cells, which kills you in the differential expression analysis, for example, but also in the clustering, they cause many problems. If you are too restrictive, it could be that you end up with a very restricted number of cells, so it is difficult then to make an analysis. So now we can use the subset function to subset our object based on this threshold, okay? And it is pretty quick. And then after this, we can also perform an additional filtering, which is the gene level filtering. What is this? We can choose to keep a gene which is expressed in at least a number of cells. And why is this? Because if we take, for example, if you keep all transcripts, so all genes that are expressed in only one cell, okay, after, when we do the statistics, we perform many tests that actually are not useful because we have information on that genes in only one cell, okay? So it kills the statistics with the correction, okay? So we can decide to keep transcripts that are expressed in at least a number of cells. So in this case, I kept transcript for which we have information in at least 10 different cells. Okay, so we extract the assay from the layer counts, which stores the count, row count of the transcript. Okay, I keep in the non-zero a logical matrix which gives me true or false. Is there at least one count? Is the count greater than 0? True or false? Okay, just to give you an idea. Hope non-zero, which is this one. It is very big, so we do not have. Okay, it's okay. This is the problem with having very large matrices. You cannot get a sense of what is going on. But still, we then extract the position in which we have the genes to keep, and then we keep only the genes which have at least one count in more than 10 cells. Okay, so okay. And then once we do this, we put everything in a new object so that we know that that object is filtered, and we can save it. And we can plot the same plot as before to see if we removed all the junk from our dataset. So as you can see, there is a lower number of cells because we discarded some cell, let's call them like this. You can see that now below this threshold there are no cells, so we successfully discarded all cells that had less than 500 UMIs. We also discarded the cells which have less than 250 gene counts, okay, number of genes for which we have the information. We have discarded the cells with less than 0.8 novelty score, and this tail that went above in the mitochondrial ratio, okay. And just to give you again the full picture, number of genes, number of UMIs, controls stimulated, and the color indicates the mitochondrial ratio. You see that we cut out all the cells that were in these three quadrants, and we only kept cells which were in the best quadrant, okay? So which had a great number of UMI, a great number of genes, and low mitochondrial content. Okay, so here we conclude the hands-on session, the first one. We will take a small break, so again 5 minutes, and after I will answer your question if you have any, and I do this just because so that you can digest everything that we saw, because this tutorial is very compact and rich of information. So I want you to be able to think about it, and if you have a question, you can ask them. Okay, so I will stop the sharing. Okay, and I will see you in 5 minutes, and we will look at the question. Okay. Okay, welcome back. Now we can see some of your questions. I saw there are some questions.
Yeah, Laura, there are some questions. Starting with Taylor, she's asking, what about using the H5 file matrix instead of TSV and MTX separated?
Okay, so you can use the unified file, but you will have to use a different function from the package in order to import. Also, the one I showed you is for the 10x genomics data, so it will look for these three data, or you can specify whether you are putting in a different input. But there are some packages designed to import other library preparation methods. So just by Googling your library preparation and import file for Seurat, it is highly optimized. So they have different packages to import it.
Thanks. And then we have also another question: where do you merge the tables from the different samples in the beginning before making the quality filtering? And could you detail this a bit more, please?
Okay, so yes, we are merging the tables just because we want to have a clear overview of the entire dataset, because then in the next hands-on we will actually merge the information coming from the two datasets so that we make the cells comparable. So we want to see whether all cells satisfy these quality controls. Okay.
Thanks. Then from Clyde, we have, are these the steps that were run to generate the Seurat object that we downloaded?
Yes. So at one point in the code, you will find one save that actually has the same name of the one that you downloaded. In that case, we will see later, I already run everything up to that point so that we can load it, because one of the analysis takes a very long time, also with not small cells. So we have 30,000 cells. So to optimize, I already run the analysis for you so that you can just load the object from that point onward.
Laura, and then from Tyler, which are the differences to be aware when plotting a log10 information compared to a raw information, and when should I use one or another?
Okay, so usually you use the log transform data when you expect to have a huge range of values. So, for example, think about some two variables that are, for example, correlated, no, in an exponential manner. So, for example, for the x-axis, you have a value of 1. For the y-axis, you have a value of 10. Then you go x equals 2, so you are here always, but on the y-axis, you go to 100. So you have very much, I also exit from the screen, but you have very much distance between your points, and you can lose in the visualization, because on the x-axis they will be very close to each other, and on the y you have this huge range. So it is useful to transform the data in the logarithmic scale so that you can visualize the data in a more compact way, let's say. But all the calculation will then be carried out on the raw data, not the log transform. Okay.
Thanks, Laura. And now last questions. What are your thoughts about using MADs, so median deviation, for setting the thresholds? And I think for when you were plotting like UMIs against gene counts, or the number of genes actually.
Yes, of course. I think that there could be some problems with using the median deviation, because if you're using the median deviation to set the threshold, you are somehow implying that your data is symmetrically distributed. But this cannot be the case for the cells. For many types of cells, you do not expect the distribution to be symmetrical around the mean. So I think that using the deviation, the median deviation, could give you some thresholds that make you lose a lot of the information in the data set.
We have a very last questions for sake of time, then we can resume with other questions if they appear and pop up in the next breaks. But actually, I think it's related to the filtering, so log 10 and row information. And Taida is asking if the filtering is made on the log 10 or the row information.
Okay, so in the calculation, we already put the log 10, and the threshold is set thinking about the fact that we transformed the value with the logarithmic scale. So the threshold is adjusted for this, but you could also calculate the row values, plot them in a logarithmic scale, and then you can choose a threshold on the raw data. And in this case, it will be, of course, a different threshold from the one I gave you, because this is already thought with the idea that is logarithmic scale, okay?
Thanks, Laura, and I think that's it for now. And there's just an additional question in the chat, like if the script will be shared, and I think we did that disclaimer at the very beginning, but yes. I mean, if you repeat it, I think that would be worth of.
Yes, all the code, so the script will be loaded in the folder just after the tutorial, so you can play with it. So it will be at your disposal.
Cool, thank you. So now I think we can resume with the next part, so enjoy.
Yes, so we can now go on. I will share the screen again, and this one. And you should be able to see the slides. Okay, so we go on with the second big part of our tutorial, which comprehends the, let's say, heavily mathematical stuff. Of course, this tutorial is tailored for beginners, so I will not go into the details of all the methods that I will explain, but I left you all the links and the details at the end of the presentation. So if you want to study more on this and go into more depth, you can do it. Okay. So in this part, we will see about possible sources of unwanted variations, the importance and the significance of normalization and integration in the data, the process of clustering, and the evaluation of the results of the clustering steps. And we will also see some hints of PCA. Of course, if you can write in the chat, if you all are familiar with PCA, we can jump that step, but I wanted to leave it here just because many of you might not know it, and it is a very important thing to, the logic behind it is a very important thing to understand better the clustering, the UMAP visualization and stuff. So once again, I put here the workflow. So we performed all the self-filtering. Now we will see all the other quadrants, okay, all the other squares, and step into the workflow. So first of all, in order to go and obtain a pretty nice graph like this that you see in all publications with single cell analysis, we have to understand that this is a plot that is somehow capturing the variation in the transcripts of each cell type and each cell. So we can have actually many sources of variation that are not actually of interest, but that they confound our data. So we will see the identification of possible sources of unwanted variation. We will see the data normalization, integration, and clustering step. Why is it important to identify possible sources of variation? Because unwanted variation. Because these sources can confound then the creation of your cluster. So as I explained you before about the cell cycle example, there are some things that are not actually the interest of our study that generate somehow noise in the background that can affect our clustering and our estimation of differences. Okay, so first of all, we have to explore our data set to detect potential confounding factors, and these allow us to determine which will be the covariates to regress out during clustering. So the covariates which effect we need to eliminate when we perform clustering. Common sources of unwanted variation in single cell analysis are cell cycle effects. So we have transcriptional differences just because they are in a different phase of the cell cycle. We have the mitochondrial gene expression, which might indicate, as we saw, cell stress or low quality cells. Sorry. And we need to eliminate and regress them out if they are not significant and if they skew our clustering in a significant way. Then we normalize data. Why is normalization important? We need normalization in order to somehow be able to compare the transcript expressions in a correct and fair manner, because and we will see a couple of examples. Sometimes we see a difference in transcript levels that is not actually due to a different transcript expression, but it is due just maybe due to technical stuff and variability, or it is also dependent on the transcript lengths, but we will see it in just a few slides. And in this case, by normalizing the counts, we can actually assure that our comparison for the differential analysis or identification of which transcripts vary more in comparison to other is actually correct. In this case, Seurat recently introduced a method that is called the SCT transform, which perform different steps and somehow correct the abundances, so the transcription levels between cells, and allow in the same step to regress out the unwanted variation that we identified before. Okay, so there is a very nice paper, and at the end of the slides, I left you all the links. It is very mathematical, so we will not go into it, but it is the one that we will use. So, as I was saying before, there are many sources of variation that makes it appear as if two transcripts are expressed at different levels, but actually they are not. One of them is the sequencing depth. So if I sequence sample 1 with 1 million read and sample 2 with 2 million reads, I will see some differences in the level of expression just because I sequenced with more reads. So here, for example, it would appear as if the green gene is expressed the double than in sample 1, okay? So this is one factor to take into account in the normalization. Also, as I said, the gene length. Why? Because just, for example, here, the blue transcript, it is shorter, so only 3 reads cover it. Whereas the red transcript, it is longer, so I have 6 reads, but the 6 reads come actually from the same transcript. So I have 6 reads here, and let's say I have 6 reads here, but they are not expressed at the same level, because here they express 2 times, and here just 1, okay? So in our case, actually we are only sequencing the 3' end of the transcript, so we do not have to adjust for the transcript length. But if you are using other sequencing methods and library construction methods that keep the whole transcript, you need to take this into account. Then, together with normalization, we need to scale the counts. And scaling, what does it mean? It means that we somehow reconduct all our counts so that they have a mean expression of 0 and a standard deviation of 1. Okay, so why do we need to scale these UMI counts? Because different cells have a varying mRNA content that can be influenced by the cell type, so just by being that cell type, and by technical variation. Okay, sorry. We are not actually interested in absolute counts, but in the relative expression level of that gene. So, as I said, each UMI is then adjusted by a cell-specific factor that is calculated. And we, in this way, we ensure that all cells have the same number of UMI counts. And this is the same principle, let's say, of normalizing different experiments, okay, just to make them comparable. And in this way, we are sure that what we find by comparing our data is true, and it is not due to technical artifacts, okay? So here, I just put a little detail in which the SC transform, which is the new algorithm that Seurat uses, employs the Pearson residuals from transformation. So each measurement is multiplied by a gene-specific weight, and the weight of each gene is estimated based on how much evidence we have that it is not uniformly expressed across cells. What does it mean? It means that if I have the information that that gene is expressed only in some cells and not in other, it means that it is important for distinguishing the cell type. Okay, so because we have a subpopulation that express it and a subpopulation that does not. So if we have more evidence that these genes are differentially expressed across cells, okay, we have more weight. In this way, if we have a gene that is expressed only by a fraction of cells, we favor it. Otherwise, if we do not imply this method, we will actually end up on pushing down, let's say, pushing down the signals given from this gene, because the genes that are actually expressed in all the cells will have more signals. Okay, so this is the trick that used in SC transformer to balance somehow this factor. So here I left just a bit of note to elaborate on what I said on the previous slide, but as I said, I will not go into it. There is a very nice paper explaining it, so I left here the reference. Then, once we normalize and scale our data, we have actually to put our data together, because they come from two different samples. So if we have multi-sample studies, the samples can come from different experimental conditions or multiple experimental, multiple batches. Maybe I performed the experiment by I sequenced it into 2 Illumina flow cells or something like this. So we have a technical variation that needs to be eliminated in order to integrate the results. The key steps of integration in Seurat is to identify shared variation. So we find gene expression patterns that are common across the dataset. We then align the cell population. So we identify, we use these genes, so these signatures, let's say, as anchors to match cells that display similar profiles, and then we merge and correct the data. And then we will see this more in detail also with the code, okay? But here I put a nice picture. So we have two different data sets. Just think about it as trying to find anchors. So these cells are similar to these cells, so I can somehow anchor them together because they are similar. And as you can see, then the result is that in each, in this case, there are already clusters. In each cluster, we have both cells from the reference and from the query. After, we will generate a graph, a plot that explain this a lot better, because we will see that when we plot them together, the controlled and the stimulated samples, we have different cluster, but the cluster is made up only of stimulated cells or only of control cells. Whereas after integration, we actually somehow link them together, anchor them together, and merge the cluster. And we see that actually it is only one cluster. And the previous result that we saw was actually fruit of technical variation, because they are different samples, okay? So, as I said, this is very important in order to cluster the cells very well. So, sorry. So, the goal of clustering is to group cells based on gene expression similarity. So we want, for example, groups in which cells that are closer to each other when compared to the rest are actually more similar from the transcriptomic point of view. Of course, the goal would be that one single cluster is then associated to one single cell type. And then we will see this also with the code. We will try different cluster resolution, is called, which somehow indicate the strictness, okay, of the rules in order to distinguish one cluster from the other. And we will have plots later that will make this all clear. And then we need to analyze and determine whether the clusters represent true cell types, or maybe we were too strict in our thresholds, and the cluster that should have been one actually end up split into two. So we have to refine our parameters. And how can we somehow cluster cells together? Seurat uses PCA scores for clustering. So again, the goal is to obtain cell clusters that are uniform, and each cell cluster should then be reconducted to a cell type. Okay, so first of all, we need to check for the unwanted variation, because if we leave it in the data, it can happen that we obtain clusters that are actually due to the variation and not due to the true biological differences between the cells. We then perform the PCR counting, and then we explore this PCA, so these principal components, to see the most driving genes and if these make sense. And then we can, based on this, assign the cell types. So if no one opposes, I will then go into a bit more detail on the PCA, just because, as you saw, it is a key step for the clustering analysis, and because I think it is very important in general for computational biologists to know a little bit how it works and how it is used to reduce the space, the dimensional space, okay? So PCA, which is the principal component analysis, is a dimensionality reduction techniques, and it highlights the variation and patterns in the dataset in a very synthesized way, I'd say. And it helps reduce the complexity of the data while keeping somehow and summarizing the key information that explain the variation, okay? Why use the PCA? The PCA reduces noise while keeping the biological information, and helps with clustering, with visualization, and in this case, we will use it to generate the UMAP plots. So, just as an example, we have here two cells, so the cell 1 and cell 2, and here we have the different expression levels of 4 genes. So A, B, C, and D are genes. For example, gene A, it is expressed at level 4. Okay, we have a count of 4 in cell 1 and a count of 5 in cell 2. Okay, and this is a matrix. I just put some values to describe these genes. If you were to draw some lines that somehow recapitulate, synthesize the most variation, where is the most variation? The most variation is between the B and C, no? It is the biggest difference. So I could draw a line in this direction, because somehow this recapitulates the greatest variation that I see in the data set. Then I also see that there is a variation in this way, because we have D, which is okay, it is here. If I consider this, D is on the left and A is on the right. It is a smaller variation, but it is still there, no? So I can just think about translating, okay, transforming these two directions to put them on the orthogonal, okay, plane. So I can put that here is the X and this is the Y, no? And this actually is PC1, which is the principal component 1, which is the principal component that summarizes the greatest variation present in my sample. And then I put them in order, so from the one that captured the greatest variation to the lowest one. So then I have PC2, because these takes somehow represent a smaller variation, okay? So given these PCs, okay, so principal components, I could somehow measure the weight of each gene on that component. So, for example, on PC1, which are the genes that contribute in a greater way to these components? It is gene B and gene C, because they are the farthest one, no? The more variable one. And in this case, as you can see, gene C, for example, on PCA1, it has a great influence. And gene B, also on PC1, it has a great difference, okay? So somehow here the influence is given by the degree of variation from the origin, okay? So here, don't get scared. This is just to explain what does it mean that it reduces the space. So here we have the cell count, the gene expression in that cell, which is this table, okay? We just wrote it in a matrix. And if we multiply the gene expression in each cell by every gene's influence on the PC, which is this part of the table, okay, we get actually the principal component score of each gene. And what does it mean? That we take cell 1 gene A times gene A PC1, okay, plus gene B cell 1 multiplied by the score, so the influence on PC1, and so on. And the same for PC2, okay? We then calculate all this. This is 4 times minus 1 plus 1 times minus 10, and so on. And we get, you see that the dimension reduces. We get a smaller matrix of 2 by 2, okay, which somehow captures all the variation that we had in the data set. And if we used this calculation and we plot the scores, we can then plot the two cells with individual dots, no? So this is a way somehow of transforming the data to reduce its dimensionality, and it helps very well to visualize our data. Okay. Then, once we have calculated all these principal components, of course, in this case, as you might notice, we have two principal components because we had two cells. But if we add another cell, we would need another dimension to here. We would need to add another axis, no, to comprehend also the expression of gene A, B, C, and D on the third cell. So as you can understand, we have as many PCs as a number of cells, okay? Okay. So in this way, we have data that is very noisy just by being the data that it is, no? Because we have sequencing, we have technical variability, and so on. We reduce the dimension, we capture significant variability, and then we use it to plot our cells, okay, and to estimate somehow how a cell is similar to another. And then, given that, we can cluster the cells together. Okay, so you have to imagine that its principal component is actually, I wrote here, a metagene, which means the result of combining many genes. And of course, there are some genes that are more contributing, so it is the influence, okay, which are more contributing to the PC, and genes that are contributing in a fewer way. But we can then analyze which gene are actually leading that principal component, that is, which genes are explaining the variability that we see in that direction, okay? So for clustering, we will use principal components, and of course, the number of principal components that we consider for clustering is very important. And why? Because if you consider very few principal components, you might miss some part of the variation, so of the biological variability that you have in your sample, and you lose the information. You do not get a cluster based on that. But if I add all the PCs, so I have 1,000 cells, I have 100 principal components, I add all 1,000 principal components to calculate the clusters. Here I might end up with overclustering, and it might end up at the extreme, one cell, one cluster, okay? Because each cell is actually a little bit different from the other, okay? So we will see also how to select the number of principal components. So once again, very great deal of information in a restricted time. So I will leave you, we can take 10 minutes this time. And of course, if you have questions, you can write them in the Q&A box, as always. And after the breaks, we can discuss the question together if we have any. And then we will go on and see and perform the code to do everything that we saw up to now. So we will see the hands-on session number 2. So here, and we'll see you in 10 minutes.
So we are now back. We do have one question, Laura, before starting with the next session and the next steps of this analysis. Is from Eva Maria, who's asking which type of normalization is better, the regular one or the SCT transform, pardon, and which one do you usually use for your analysis? I think, Laura, you're on mute. So now we can hear you.
Actually, during the analysis, we will employ both normalization, but actually the regular one we will just use to explore the data and see whether we have some unwanted variations, okay, in our data. Whereas when we actually go and analyze the data, we will perform the SC transform. So we use normal, let's say classical normalization to identify the factor that needs to be regressed out using the SCT transform. Okay, so let's say that for actually the true analysis part, it is always better to use the SCT transform, because otherwise the signals coming from genes that are lowly expressed or are not expressed in many cells are actually suppressed somehow, are penalized, okay, in the weighting. So for the true analysis, we use SCT transform, but to explore the data and understand a little bit, we use the classical normalization.
Thanks. We just received a second question following that. So is SCB like SCRAN or SCRAN? And is the default normalization you are talking about the same as shifted logarithm? And with default one, it means Taila means.
Okay, no. It is different. So the SC transform, as I said, it is actually two different types of data steps that are performed. So we have a weighting step of the genes which are weighted, as I said, based on the Pearson residuals, so also based on their variation, and the weight are adjusted to favor lowly expressed genes or genes that are expressed only in a subpopulation of cells. And then the second step of the SCT transform algorithm includes the regression of unwanted covariates. But yeah, just for the mathematical details, I send you on the paper. It is very clear and detailed on the mathematics used in the algorithm. So if you're interested in that, you should totally check the paper. So, okay, so do we have other questions?
No, I think so far that's it. I was waiting for any possible reply, but at the moment, nothing has popped up. So I think we can move on, and then eventually we can resume other questions after this session. Great.
Okay.
See you later. Enjoy.
We will now go to the second hands-on. So I will share the screen if I can. Okay. And this, share. Okay. So you should see now the RStudio environment, just where we left off. So we have now the second part of the hands-on, where we will perform all the steps that we saw before. So we will check for unwanted variation. We will perform PCA calculation. We will choose how many principal components to use for the clustering. We will perform the clustering. We will check somehow the quality controls of our clusters to understand whether our clusters satisfy us or not. And then we also end up and assign cell types based on the expression profile of each cluster. So, first of all, as I just said, we can try and understand which are the sources of unwanted variation in our data. And to do this, we can apply the simple normalization. So there is a function in Seurat, which is the normalized data. So we work on the filtered Seurat. So the data set that we already filtered based on our quality controls. And we run this for the normalization. It is usually fairly quick, but if you have a great number of cells, so maybe 100,000 cells, keep into account that these steps will be very time expensive and also RAM consuming. In this case, we use the simple normalization to explore the data, and then we will apply the SC transform to actually then work on our data. So we normalize our count, and then we explore the data to check if we need to correct the data somehow. And what does it mean? It means which covariates do we need to insert in the SC transform to regress out. So one of the unwanted variation is the cell cycle, as we saw. So first of all, we need a file with the markers of each cell cycle. This is available on GitHub. So we have genes that are highly expressed in each phase of the cell cycle. Okay, so I will read, and we can see. Okay, so we have the phase, the gene ID. As you can notice, they are ensemble gene IDs. This is just the date from the last modification of these markers. So then we will connect to the annotation hub, which allows us to connect and get annotation information on this gene. So we load the annotation lab, okay, library, and we activate it. Then we access the ensemble database, okay, for the organism Homo sapiens, and we collect the information. And how do we do this? We have actually many updates in these annotations. So in this command, we actually get, and I will show you, the code for the latest annotation. So it is the most updated, no? So we get our query. We queried the ensemble database for the Homo sapiens, which gives you a list of versions of the database, and then we took the last one. Okay, so it is the most relevant. Okay, the updated version. So then we can query it. So we go to the annotation hub and we extract the annotation relating to our ID, which is the latest one, okay, which is this, and we save it. Okay, then we get the annotations and we transform these what I called ADB. If you look up here, you see that is an object, a particular object. From this object, we can extract the information in a data frame manner, okay, in a data frame structure. Okay, maybe I can lower this one so that you can see better all the, okay. So now you see the annotations are here, and it is 87,000 observation of 13 variables, okay? So we have the gene ID, the gene name, we have the biotype, we have the position in the genome on the chromosome, if it is the positive strands or the negative strand. We have the information about the product, so we have the description part, and we have the gene symbol, the entrance CD, and stuff like this. From this, we do not need all this information in our case. So from this, I will extract just the gene ID gene name, the sequence name, gene biotype, and description. Okay, so now our annotations looks like this. Okay, we just extracted the information that are important for us. And then we can merge these annotations with the list of cell cycle genes that now I will show you. Okay, so the cell cycle genes, as we saw before, we have the phase and the gene ID. The gene ID is also present in the annotation data frame that we created, so we can use it as key to merge the two data sets. Okay. Okay, and now if I check, I took the wrong object. Okay, if we check the merged data frame, we see that we have the information of the phase, but also all the annotation that we had extracted from the annotation hub. Then we can filter and make the data frame specific for the genes that are that characterize, that are biomarkers for the S phase or the G2M phase. So we extract the S genes and the G2M genes. And just to give you an idea what we are doing, okay, we extract the name. So we filter for the phase and we pull the gene name. And then we can somehow give a score, assign a score to each cell that tells us in which phase it is more probable that this cell is. Okay, it gives us information on the state of the cell. Okay, so here we have a function that uses as input the Seurat object that we are working on. And then here we give the features, so the markers of the G2M phase and the S phase, so the one that we generated here. Then, okay, it gives us some information that some genes were not found, but it's okay. And now we can see that if we pull our metadata, together with all the information that we computed before, so mitochondrial relation, stuff like this, we have a score for each phase that we analyze, so the S phase and the G2M phase. And in this column, the column phase, we have the phase in which the cell most probably is. So given these two list of biomarkers, which one describes better my cell? Okay. So now that we have this information, we can perform we can find the most variable features, and we can scale the data in order to calculate the PCA and see whether the cell cycle influences the cluster and the distribution of my cells or not. Okay, so with this, okay, I will launch it because some of them require some minutes to run. With the find variable features, we find the most variable genes. In particular, we have to specify the selection method, which is the VST. There are many methods, and in the Seurat manual and tutorial created by the developers of Seurat, they are all very well described. The VST method is the most used, so we'll use this, and it is the one that is suggested for analysis. And in particular, we use this method to select the first 2,000 features that are most variable in our data set. Then we scale the count. So we simply with the function scale data, okay? And this scale our data, so makes it so that we have a mean expression of 0 and a variance of 1, okay? We can see, for example, the result of the variable genes. For example, we can rank them, okay? Okay. And we can see the top genes, okay? But if we want, we can also plot them so that we have nice pictures. Okay. Okay. And as you can see, the 2000 features that are most variable in our dataset are indicated in red, whereas the non-variable features are indicated in black. Here we have the average expression and the variance on the y-axis. And as you can see, all the variable feature that we identified are actually highly variable, no? They represent the upper part, no, of the variance, okay? Here we have labeled the most variable, okay, once. And this is always a nice plot in order to see whether you are selecting too many features or too few features. Because based on the cell types, it could be that by selecting the most variable, the 2000 most variable, you maybe select only this part, so you lose some variance. So in this case, you would need to increase the number of genes that you consider in order to somehow get all the most variable, okay? Then we perform the PCA. And in order to do this, of course, we consider the most variable features. Why? Because with the PCA, we are trying to summarize the variance of our dataset. Okay. Then, after this step, so now it is calculating all the principal component scores, okay, of each gene expressed in each cell. And then we will plot the PCA. So here, as you can see, it gives you some information. You see PC1, positive and negative, and these are all genes. These are the genes that are most contributing to that principal component, okay? And here it prints out the first 5, okay? But there are many more. We can then plot the PCA, and we group the plot based on the phase. And here, what are we seeing? On the x-axis, we can see the principal component 1, and on the second, the principal component 2, which are, again, the principal components that summarize the most variability in our data set, okay? And in this case, we have the G1 phase, the G2M phase, and the S phase. If, in this case, you see all these graphs, all these PCA plots are very similar to each other. If instead we would see somehow some clustering that is linked to the phase. So for example, if we saw G1, so pink only here, green only here, and S only here, it would mean that the cell cycles actually have a great effect on the clustering of our cells, because it influences a lot the principal components. So if we would see pink, green, and blue, for example, it would mean that we would need to regress this out from our data, because it is influencing a lot the distribution of our data. In our case, as you see, there is actually no difference between the phases, okay? And what does it mean? That our data set is not influenced by the cell cycle state of our cells, okay? So we will not regress this out. Then we can check for the mitochondrial expression effects. And again, also here, I report that here, if you see differences and you would not expect them in your data, then you will regress it out. But if you see differences, but the differences are explained by the cell types you are analyzing, you do not want to regress this out, because it means that you will lose meaningful variability in your data set, okay? So be careful of always having expectation for the data set you are analyzing, okay? So in order to do this, so it is the same thing for the mitochondrial expression. We will somehow categorize our cells for the mitochondrial transcript content. So we use the summary function on the mitochondrial ratio calculation, so that it gives us quartiles, okay? And then I will, just for the sake of this analysis, break our data and label it so that everything that is lower than the first quartile is low mitochondrial ratio. Then we have medium, medium high, and high. Okay. Then I perform again the plot, but I group it by the mitochondrial, this one, the mito-FR, which we have now calculated, so which are the ranks somehow, okay? And here we actually start to see some differences in terms that, for example, if you compare the pink, sorry, the pink PCA, so the low content, with the high content, we see that here we have much more cells contributing to this cluster than these cells, okay? So this could actually mean that this is a source of variation that we do not want, okay? Because actually, as we lower the mitochondrial content ratio divided by class, okay, we see a greater contribution of cells to this cluster, okay? So these clusters are actually driven by the mitochondrial content. So we will remember this for the SC transform, okay? So we will regress this out. Okay, so I will just drink a bit of water. Perfetto. Okay, so as I said, now we will perform the SC transform, but since our data came from two different samples, we have to perform the transformation on each sample individually, and then through the integration and the anchoring steps, we will merge them together, okay? So for now, we have to split the object, and we split it by sample because sample was control or steam, which is stimulated, okay? So then we split the object. Then for this step, we have to change a little bit the memory that is allocated for object loading, okay? Because we know that this could be computationally demanding. And now we perform the SC transform. So this actually takes a bit of time, not too much because we are not working with many cells, but I will get this chance to just take a couple of minutes of break so that I can fill up on water, and I will see you in just a couple of minutes when this process is done. Okay, we are back since it's finished computing. So once again, I share my screen and we can continue with our programming. So as you can see here in red, okay, these are all the steps it performs. So we have different calculations, okay? Then we have the regression of our Mito ratio because it was what we told the algorithm to regress out, okay? It is calculating the residuals, it is centering the matrix of the counts, and now we have set the default assay to the SCT calculations, okay? This will be very important later on. So for the clustering, you use this. But then when you are estimating and evaluating expression levels, you do not use these transformed values, okay? Okay. So here, okay, I wrote, okay, that the default assay is set to SCT now, but we can view actually the various assay that are stored into our Seurat object. And as you can see, we have an RNA level which have raw counts. And then we have the SCT assay, which is the SCT transform values, okay? Again, since this calculation might take a while, I actually put it here strategically to drink a bit of water, but always remember to save everything so that you can load it and you do not have to run it again every time. Now that we have transformed our data for each of our sample, we can now integrate it. So we can find the anchors and integrate the information in order to put the cells on a comparable, let's say, plane, okay? Then, here, just a note, do not perform the integration just by default, but always analyze the data to see actually if the integration is needed or not, okay? So, okay. So, we now plot the UMAP, which is that pretty plot that we always see. So we perform the calculation. And as you can see, we need to give the reduction, and in this case we give that our dimensional reduction is PCA, and we give how many principal components do we need to take into account to somehow visualize this on the UMAP. And I selected 40, but actually it can be any number, and we will now few minutes see the choice of this number. But these I just put 30 just because we want to see how the data is put. So then with the dimplot function, we actually plot the UMAP, and here it is. The UMAP, it is a bit complicated, but it can help just to think of them like they were principal components, okay? It helps your reasoning if you put it like this, but they are actually a bit different mathematically speaking. But here, what is the meaning of this? Each dot represents a cell, and cells that are close to each other are very similar from a transcriptomic point of view, okay? Here I colored the control in pink and the stimulated sample in blue. And you can see that somehow there are a similar cluster, but they are like mirrored somehow, no? And if you would perform a cluster analysis like this, you would end up with one cluster here made up only of stimulated cells, one cluster here made up of only control cells. But you do understand that this actually is due to technical variability. It is not true biological variability, because when we will anchor and perform the integration, these would actually fit together well, okay? So here I put the code to perform the integration. So we select the integration feature, and in this case, I selected the 3000 features, okay? These are fairly default numbers I put in here. And they work well with the great majority of datasets, okay? But of course, you can feel free to experiment with different degrees of threshold, no? Then we have a function to prepare the integration, and we actually use as anchors the integration feature that we selected before, okay? And then we actually find the anchors in our normalized data. And finally, we perform the integration. Okay, all of this takes a bit of time. It takes, for this data set, if I remember correctly, it is around 20 minutes, okay? So this is the reason why I put the already saved object in the folder, because if you want to play around, you can just load this elaborated data set already integrated. And now, as you can see, it takes a bit of time to load the objects, and this dataset is also made up of not too many cells for a single cell experiment. It is just 15,000 and 15,000. So you can imagine that when you load a very heavy dataset, like for 100,000 cells, it takes a bit of time just to load in the data. Now, again, we run the PCA on the integrated object this time, and then we plot it to visualize the data. So somehow, how is UMAP better than the principal components plot. It is better in a way that when we are plotting the PCA, we only plot the first 2 dimensions, which again, they are the dimension which capture the most variability, but we can only see the 2. Whereas the UMAP actually gives us information and summarizes the variability coming from different principal components. Okay, so again, we perform the PCA, so it prints out the first 5 PCA, the leading genes, the most contributors, okay, for each PCA, principal component, and then we can plot our PCA. So you see, actually they are pretty similar, okay? Of course, we are only watching the PC1 and PC2. So we can see now that the distribution is fairly similar among the control and the stimulated. We just see a little bit more dispersion, if we want to call it that, up here, okay? But now we can try and plot the UMAP. Here, just an advice. Since the UMAP is a stochastic algorithm, it can be that each time you run it, it gives you a little bit different representation of the clusters. So it is always a good idea to set a seed, as you can see, so that once you set the seed, every time you run the code, you generate the same UMAP each time, okay? So here, again, just for the sake of visualization, and in this case, we are checking whether the integration of the data set worked, okay? So we will now see actually if the 2 representations on the UMAP are somehow, I should do like this, are somehow comparable, okay, if the integration benefited our data. Okay, and we plot. Okay, so as you can see, if I have it, okay. Before the integration, we had somehow this mirrored kind of clustering, okay. Whereas after integration, we have a superposition of the cells in each cluster, no? So each cluster is actually made up of cells from the control and the stimulated. Of course, it can be that there is a cluster made up only, for example, of stimulated cells. And this actually could represent a cell type that maybe is not present in the control. I imagine like activated T cells or something like this compared to non-activated T cells, no? So it can be that there are some clusters only made up of this, but it is not possible that each cluster is made up of only cells from the controls and only cells on the stimulated. And okay, here I also put the code to do a split, just because when you have many cells, it can be that in this UMAP you only see the sample that is plotting last. As you can see, the pink is always background and the blue is always foreground. But for example, if you have many samples, it can be that you do not see actually the distribution. So it is always better to plot them also split so that you can see whether these clusters actually have more or less the same contribution from each patient. So this UMAP, of course, is only to check whether the integration was successful. Now we have actually to perform an UMAP that will be used for the clustering. So in this case, we need to decide how many principal components we use to calculate the UMAP. So first of all, we can explore the principal components. And this is particularly useful also when looking for unknown cell types. Because if we see, for example, a cluster that is described by one component that is led by particular genes, that is a clear indication that maybe that is a subpopulation of cells. Okay. Here, with the dim heat map, I plotted the first 9 principal components, and they are heat map in the first 500 cells. Okay, so in each heat map we can see which are the leading genes, which is somehow the kind of the same of this. Okay, but in a graphical point of view. In this case, you could plot also 100 principal components, and you could choose to include in your UMAP calculation for clustering the principal components up to the component which becomes all noisy, let's say, because you see that the first is very clear, the distinguish, the delineation of the two positive and negative. So you have one cluster, this positive and this negative, and all the other in the other cluster. You have to imagine it at one end of the axis and the other end of the axis, okay? But then just also at the 8th, at the 9th, you see that there is somehow more noise, and it is more difficult to differentiate the clusters, no? Of course, if you end up going through hundreds of these, it is somehow pretty subjective and difficult to choose the point at which this principal component is noisy, I wrote here fuzzy, or not. So we can help ourselves also with our other methods. For example, we can print out the first 5 positive and 5 negative genes contributed to the principal component. And in this way, we could, if we know our cell types very well, we could somehow get a sense of whether these principal components actually are capturing a kind of cells compared to other kind of cells, because we have, for example, some markers. We have the GNLY marker. We can have, for example, the CCL, which are markers of precise type of immunological immune cell. Sorry. And then we can also plot an elbow plot with our principal components. And what is this elbow plot representing? We have on here the number of the principal components. And here we have the standard deviations of our dataset that are contained, summarized in our principal component. So as you see, they are always in order, because the first one contains summarizes the most variability, and then we have all the others. And this can help us in deciding whether at some points adding more PCs, so more principal components, does not help us very well in capturing variation. For example, if we pass from one principal component to 2, we have actually all these standard deviation explained more than if we only consider the fourth principal component. So we have here that we add a significant explanation of the variation in our data. Whereas if we come, for example, here, you can see that by adding these, I don't know which, 39, 37, 38 principal component, we only add this much of the standard deviation of our sample, okay? And here I only plotted 40, but we can go up to 100, for example. And you will see that these always become smaller, smaller, and smaller, okay? So in this case, you can somehow set up, let's see if I can, if it doesn't take too much time. 50, because I calculated 50, it's okay. But you see that it decreases more and more, no? Then, given this, usually you can choose the point in which you say, yes, well, by adding these the next principal component, I do not get much of information, so I cut it, and I consider this. For this example, I choose 40, but I recommend you to try also different thresholds, so that when you try different thresholds, you end up, and then you perform the clustering. At one point, you perform a clustering that is not actually any different from the point before. So including those extra PCs that increase the computational time, it is not actually beneficial at all. You are only increasing computational time and memory just to compute the same thing. So then you cut, let's say, your PCs there. Here I selected, as I said, 40 PCs. In this case, we use the find neighbor function. With the parameter dims, you indicate how many principal components to select, and what is it doing? It is performing a clustering based on the k nearest neighbor approach. Okay, so that it finds the neighbors, and I can run this in the meantime. It can find neighbors based on the graph that comes out when you plot everything using the information coming from the 40 principal components. Then once we find the neighbors, we perform and find the cluster. So we perform the clustering, which is this, as you can see. In the Seurat manual, there is explained very well all these clustering method, because they inserted them in the package. Okay. So, perfect. Just one thing that I should say. The resolution parameter is very important because it decides, as I was telling at the beginning of the tutorial, the granularity of the clustering. So the constraint to say that one cell belongs to one cluster or another, are they relaxed or are they more strict? Do I end up with just one single cluster encompassing all cells, or do I end up with one cell, one cluster? this is the parameter to set. And as you can see, you can collate many different resolutions. So this function will calculate the clusters at resolution 0.4, 0.6, and so on. So we can see, okay, it is just another way of seeing. Okay, here we have some numbers. The numbers indicate the cluster, so cluster 0, cluster 1, and we have for each cell the cluster to which it belongs. And sometimes the clusters change with the changing resolution. So, for example, we have 2. The first cell belongs to cluster 2, resolution 0.4, at cluster 2 at 0.6, and the cluster 1 at 0.8. Okay? So, my advice, I know it can be very tedious and difficult to choose, but it is always best to go through and plot and see and check all these resolutions, and then you choose the resolution based on the clusters that make more sense at a biological level. Okay. So, for example, here, let's say, and okay, here I just put that it is worth exploring between the 0.4 and 1.4. Of course, you can explore also a wider range, but I mean, it is usually more work, but in my experience, this is the range in which you will find your true clustering. Okay, so now I just said, okay, I choose as cluster the cluster generated by the resolution 0.8, and I plot the UMAP. As you can see now, the UMAP is the same as before, but I have different colors for different clusters, okay? We can do the same thing for just to show you how this changes. This is the clusters with the resolution 0.4. As you can see, here we have many clusters, and here we have less clusters. For example, this part of the graph, here with a higher resolution, 0.8, we have the cluster 0, and here the cluster 6. Whereas here, we only have one single cluster. So the lower the resolution, the higher chance that all of this is one cluster, let's say like this. Then we can perform some quality controls to check whether we performed our clustering well, and this is also useful to choose the resolution at which to cluster. Okay, so by doing this, what am I doing? I am plotting the number of cells belonging to each cluster. And this is a good measure to see whether there are clusters with a low number of cells or clusters that are composed only of one sample. This is another way of representing this. So these are the UMAP divided by the control cells and the stem cells. So we can get it in a more, let's say, overview way. I put here different representations so that you can choose and explore which one is better for you, but these are all representing the same thing, okay? Okay, this is another quality control. In this case, as we did before, I divided the cells by the phase. And if you remember, we were good on the different cell cycles, so we did not have to regress it out. And it is confirmed by our clustering, because the clusters are not driven by the phase in which the cells are. Okay, this takes a bit of time, but with the feature plot function, you can plot UMAPs that are colored by a parameter that you have in the metadata information. So, for example, here I plotted all the sources of possible variable information, and as you can see in blue, there are the cells that have the highest, let's say, number of score for each of these parameters. So this allows us to understand whether some clusters actually represent one of these confounding factors. For example, if the mitochondrial ratio, we had to regress it out because it was actually influencing our data a bit. If here I find one cluster that is completely blue, I have the suspect that that cluster is not really good because it is influenced by the mitochondrial ratio. Okay, this is the meaning of this plot. Okay, then here I also put some codes. These are all graphs that helps you understand your clusters. Okay, these represent the number of genes expressed in each cluster, and it can give you an idea of the richness of the transcriptome in each cluster. And if we see any differences in any of these plots that are driven by unwanted variation, this is the time to go back and refine the clustering, or we need to add some covariates to regress out. Okay. Then, okay, as I said, we can then go on and explore which principal components are driving the different clusters. And this is very useful to understand cell types. Okay, and this is just the code to plot. Just give it a minute. To plot the markers on the UMAP. Okay, so here we have the contribution of each principal component on the UMAP and on the cluster. So it is the same idea as before. The more blue, more contributing. Okay, and for example, in this case, we can look at here, principal component 2. We have here a cluster number 5 that has a great contribution from principal component 2. So we can, for example, print the leading genes, okay, for these principal component. So cluster 5, which is highly driven by the principal component 2, has as greatest contribution these genes. And if I know my cell types very well, I can say that the GNLY and the NKG7 markers are actually corresponding to the NK cell types. Of course, this works very well if you are a super expert of your cell type, but this could also help you identify rare or unknown cell types. So inside your cluster, you see a small subpopulation, very blue. Okay. So, okay, here I just put some code to plot, okay, different cell markers. So if you know the markers of your gene, like the PBMC genes, the PBMC cells, they have very well-known markers, the immune cells. So for example, if I want to identify the CD14 plus monocytes in our UMAP, I can see which of these cells express CD14 and LYZ the most. These are probably the monocytes. And I put here some example with different cell types. But I also want to show you how to use this tool, which is called the SCtype, which is of very great help when you are not actually an expert of your cell kinds. And this is a tool that, given a set of genetic marker, both positive and negative, it has many cell types already available with all these markers, and it automatically calculates some score that is somehow a probability that that cell is of that cell type, given the transcriptional profile, and assigns the cell type automatic. Of course, if your cell type is not present in the available ones, you can also give it your set of biomarkers. Okay, so if you are, for example, interested only in some particular subtypes of cells. So this is all code that is available in the tutorial of the SCtype tool. So it looks a bit complicated, but actually it is all explained very well in the tutorial. And you can find it on GitHub. And it calculates the likelihood of each cell belonging to each cell type. In here, I selected the immune system cells to annotate our UMAP, and I just said that the low confidence cluster, if you are not very sure, don't give it a name, just mark them as unknown, okay? And here we have finally our UMAP, as you can see, with our cell types annotated. And all these cell types were present in the database with the immune system cell markers. Of course, these can be useful and helpful, but if you're looking for rare or unknown cell types, you need to explore manually which are the genes leading or the principal components leading that subpopulations, that cluster. With this, I end the coding part. We will take just 5-minute breaks. Then I will answer your question and we will close the tutorial. So see you in 5 minutes. Okay, so sorry for the little delay. We are having just a bit of a technical issue, but we are now at the end, so we can just go on. And I see that there are some questions. I will try to answer them in a very summarized way. So we have 2 questions. Should we always perform the cell cycle step, or are there cases where we should not perform it? I'd say that it is always safe to perform it because you do not know a priori whether your cell are more in a cell cycle state compared to the other. So it gives you a good sense of unwanted variability. And also it does not take too much time. So I think it is a good quality control to have. Then we have why 2000 genes? How should we choose the number of genes in the function variable feature? About the variable feature, we can use the plot that we plotted before, which is the variable feature plot with the red and black dots. So we had the variability of the genes. So we had the most variable genes up. If you remember, it was like a cloud. That plot is basically the plot that helps you in understanding the threshold of 2000, because if selecting 2000 you select all the transcripts, it is not good. You need to find a threshold which selects the most variable, and by plotting that you can somehow assess it. Then I saw that Mirko is back. Okay. So if you wanted to go on to the questions.
So yeah, sorry Laura for the technical issue. Sorry everybody. Yeah, I think you answered both the answer from both the question from Eva Maria, right?
Mm-hmm.
Cool. So another questions from Eva Maria is, have you worked with Harmony to perform the integration, and or do you recommend it?
So I personally do not have worked with it, but I know many people who have, and it is a great tool. In my case, I didn't need to perform very complex integrations, but if you have many factors relating to technical variability, I actually advise you to use a Harmony because it gives you better, let's say, power or control on the integration step. So yes.
Well, thanks, Laura. And then again, does UMAP need the seed, or should we use it in more steps?
No, it is just the UMAP because that is stochastic, whereas all the other things usually gives you the same result.
Well, thanks. And then I think it was during your script, but eventually I'm gonna ask Tyler to add any other details on that. But like she asked, are these cells randomly distributed outside the clusters, low quality, or are them how in a cell type transition on the principal component analysis? I don't know if that's way some sort of difficult, so out of context. So in case it is, I would ask Tyler to be more specific on that questions, please.
Yes. So.
Yeah, and I think we can move forward with the other question. In the meantime, we're waiting for more details on that. And like, why don't you use clustering to see the visualization of the clustering?
You can use it. I mean, it is another way to evaluate everything. The thing for me is that with a single cell, actually the class tree, I always find it a bit difficult based on the class tree to help me choose. So usually I just go on and plot, for example, the features, the PCs that drive the clustering and so on. But it is always a good idea. It is just another way of looking at the data. So you could do it.
Cool, thanks. And then from Clyde, does SC type work for non-human data sets?
No, they already have annotation regarding humans. But for example, I used it analyzing mouse data. And I did it by taking the markers for each cell type from the mouse atlas. And then I just generated, it is basically an Excel file, okay, just a table with the positive and negative markers, and you can load it in the SC type and perform the annotation on your based on your markers.
Thanks. And then, like, Arya is asking a more kind of syntax-related questions on the terms marker and DEGs, so differentially expressed genes, can be used interchangeably when we compare to clusters.
So, I'd say if we are only comparing two clusters, then yes. But you would need to make sure that when you perform the differential expression analysis, if, for example, gene 1 is upregulated in cluster 1 compared to cluster 2, you would need to be sure that that gene is expressed in each of the cells of the cluster in order for it to become a biomarker of that cluster. But yeah, basically the idea behind the assignation of the biomarkers is that it is expressed in that cluster, in the cell in that clusters, and not in any other cell. So.
Thanks, Laura. And I think that from Tyla, that the question she was asking, she's saying that it would be easier with the figure. So I don't know if it's easier to get the figure actually that she was referring to. Otherwise, I think we might be in contact with you
so just because we are a bit short on time, I'd say that if you want, you can write me an email. I think the contacts are in the tutorial. Otherwise, it's just surname.name at I can write it if anyone has a question. It's a serb.it. So if you want to write me any questions, as soon as I have some time, I can answer you. Okay, so if we have no other questions, I will very briefly go over the last part of the presentation.
Yeah, so far no other questions. So
okay, so I will share the screen and I will try to end in max 5 minutes so that you are not late for the rest of your day. So you should be able to see the slides. So this again is just the workflow, and we saw basically all this part. Now I just will show you some possibility for downstream analysis. And here I put all the resources that I used to perform and to learn about single cell RNA sequencing analysis. And I think they are very great, so I left them here for you. Here, just for the downstream analysis, and just to give you an idea, we can perform a pseudobulk differential expression analysis. What does it mean? For example, in the dataset that we explored, we had stimulated against control cells. We could, for each cell type, so for each cluster, we can somehow aggregate the counts of each cell in order to have a pseudo bulk in that cluster, and then perform classical differential expression analysis, for example, with DSEC and stuff like this. And we can see whether the stimulus has changed the expression in that cell type. So, for example, here it was a heat map I plotted, and these are 81 cells from the mice lungs, okay, divided by genotype. And you can do all the analysis you can do with the classic bulk RNA-seq. Another kind of analysis could be the differential abundance analysis. So you can see if, again, here is a genotype, but it can be a stimulus, for example, if one cell type is enriched after a stimulus when compared to a control situation. Here, for example, then you have to perform the statistical testing, but in this work, I remember that, for example, the B cells were more enriched in the wild type samples than in the other genotype. As I said, you can explore the cell population markers. So now you know everything about these plots. So the UMAP, this is the loadings of the PC, of the principal components. You know how to plot these. These are just other graphs to show the functional information of these cell types. And this is actually a plot that we have not seen, but it is readily available in any of the manuals I linked you. And this is a violin plot representing the distribution of the transcript in each cell type. So of this gene in each cell type, how it is the distribution of the expression. And these I just leave here for your knowledge. These, the pseudotime and RNA velocity, are fairly novel analysis and calculation you can perform on RNA-seq data. Actually, if you want to perform this kind of analysis, you also need to have it in mind when you set up the experiment, because in one case, the RNA velocity case, you have to have information on the spliced and unspliced RNA data. So you can maybe choose a library that allows you to see that. Okay, and these are just the very superficial points of this analysis, but I leave it here to just give you an idea. And with this, I conclude with these two QR code. You can, on one hand, go on the page of Professor Malerba Lab, because this project started when both me, Mirko, and Professor Malerba were working together in Verona, and now we are a bit across the world, across Italy. And here I leave also a QR code for the feedback, because this is our first time performing a tutorial on RNA-seq analysis. So we would like to know how it went, and if we can do something to better ourselves and the tutorial. So with this, I end the sharing, because I want to see you, okay? And I say happy continuation of your night or day. I don't know, based on in which part of the world you are. And thank you for staying with us until the end. So Again, for any question, you can write me, and then also I can let Mirko and the professor know. So have a nice rest of the day.